And then I've probably said what I need to about the in vivo translation, but there's you know, circumstantial evidence that some cases these phenomena that you measure in vitro translate into the in vivo situation, and other times they don't, and we not have a very good understanding of exactly what the phenomena are that are complicating that. So you can see from that really what we want to do is to be able to design the compounds appropriately, know about it in a sort of meaningful cycle time, and then have confidence that those effects will translate through into a real system. So the slide on the expected impact, I suppose this is a kind of real future state vision, but we expect that compounds with optimized kinetic profiles, and I guess that doesn't necessarily always mean slow off rates. There may be times when rapid off rates are desirable. Would be more likely to succeed in development if that understanding is factored in, and hence you would reduce development attrition. That's as maybe, but I think that needs to be coupled with the ability to bring that about by design, so that you increase the number of compounds that have the appropriate kinetics that you can progress into development. So in terms of the public-private collaboration, it's interesting that when we first kicked this off, obviously we had a few discussions with various industrial partners, and what was interesting was that everyone seemed to describe the same kind of situation where the companies seem to think this is really important, but have only applied the techniques in a sort of sporadic way. You know, one or two projects have looked at it, but not consistently across projects. And that knowledge has maybe not been disseminated, quite often been kept within the projects or within the companies, or not exploited in the best way. And I think most of that comes down to resource. That I think tackling this wholesale is probably too big an issue for any one company or company in collaboration to take on their own. So I think that makes it ideal for this kind of situation. And we hope that by pooling our expertise and our experiences in that we can provide complete and consistent data sets that we can make available for future study with collaborators. Obviously, the other thing is to bring in, this is a real kind of multidisciplinary field, so we'd have expertise from structures, from biophysics and pharmacology, as well as chemistry to help uncouple this problem. The plan is that we'll make available tool compounds, assay reagents and what have you for the consortium to study. That needs a bit of work to define exactly what it is, the exact nature of those, but that's the plan, is to work that through in the time frame that the academic consortia will form. And hopefully this is something that's of interest to collaborators. I'm sure that it is. I know there's lots of people that do work in this area already and are interested. And I think, I would guess that they probably suffer from the same sorts of frustrations, that the systems and the tool compounds to study aren't necessarily readily available. And hopefully we can address that via this project. So this is intended to be entirely pre-competitive. So what we need to do is identify systems that the EFPIA members are prepared to share freely, which probably means very well established old targets or maybe dead targets. Now that needs a bit of further discussion really. I don't want to commit too much to that at this stage. You can imagine the legal wrangling that will go on. I'm not looking forward to that discussion with the company lawyers, but I think what we do have is a commitment to sort it out, and we will find a way to make that work. And the plan is to make the data we have on those available to everyone, potentially through a database that everyone could access. And where there's gaps in that data set, then we will intend to plug those with experimental work as well. So the intention here is that the deliverables should be generally relevant, so they're not specific to individual targets. So we want general rules that everyone can apply in future drug discovery projects, and we're not looking for new ways of breaking the targets that we're studying, because obviously that leads to further complications in terms of intellectual property. So that makes it critical that they are targets that are definitely of no longer interesting if we can come to that conclusion. Obviously, there may be some ways to make that work in other areas, but the IP around that is much more complicated. So in terms of the makeup of the project, what we'd like to structure this project in a way that tackles the three areas that I've outlined, really. So there's three separate work packages in each of those three areas that we envision bringing through. And I guess the relative emphasis of these is subject to the proposals we get and the interest we get. It's not essential that this is a kind of equal split between them. We might find that some are more tractable, some are more interesting. It's not essential that we tackle them all with the same kind of level of effort. But I should stress here that this is not intended to be prescriptive. So I think everyone will hear the problem statement. There are other solutions. This is just the way we kind of broke down the problem and structured it. So other innovative proposals that tackle this from different angles, we'll welcome those kind of suggestions as well. We just don't know what they are yet. So in terms of the applicant consortium, like I say, we'd envision, with that caveat that I just described, that we'd get proposals in each of these separate areas and potentially just link these over together in a kind of umbrella organisation. Obviously, where there are links between the two, we can exploit those, but I guess really we probably envision these being separate pieces of work. But with the consortium in place, we intend to coordinate those studies and make sure all the knowledge is shared and transferred within the network. So what we were kind of looking for in terms of molecular understanding is, I've probably said this already, but a determination of what the individual contributions are to on and off rates, which allow an analysis that uncouples what general considerations there may or may not be for those things. And that could be a kind of whole molecule level in terms of thermodynamic contributions, but it also could be specific functional group interactions. So one of the things I'm quite interested in is to understand whether, for example, an acid-base interaction leads to a much more general difference than, say, a hydrogen bonding interaction or a lipophilic interaction. And because at that level, then you could start to understand how you might be able to design these properties in and out of molecules.